Hello friends, welcome back to my channel Plant Pathology Pathsala. In this video we will deal with the fungal mediated transmission of plant viruses. So first about the history part, the fungus as a vector for the plant viruses was reported by Teakle 1960. In the previous video we have discussed about the nematode mediated plant virus transmission, and the nematode as a vector for plant virus was reported firstly by Havitt et al. 1958. So remember these two names. These two names are very important, very frequently asked in different MCQ based questions. Okay, so Teakle et al. For Teakle 1960 for fungus mediated transmission of plant virus, and Havitt et al. 1958 for nematode mediated fungal transmission. Next, the fungi act as vector for several soil-borne viruses, just like the nematode. Here the viruses are soil-borne in nature because the fungi is here. The soil-borne fungi only can transmit the viruses through the Jhumishree. That's why this viruses, specifically the soil-borne viruses, are transmitted by these fungi vectors. And this the virus transmission occur mainly through the Jhumishree. Okay. Here it may be associated with Jhumishree internally or externally. Okay. So mostly the Jhumishree producing fungi are the plant virus transmitter. And which fungi generally produce this Jhumishree? Obviously the lower fungi, because Jhumishree are motile asexual spore. Okay, and motile asexual spores are produced by lower fungi. In the higher fungi, motile spores are absent. And the first reported fungi which is transmitting the plant virus was Olpidium brassicae. This is very important, and the first plant virus that was reported to be transmitted by the plant virus that transmitted by the fungi is lettuce big vein virus. Okay, so remember these three terms, these three things, that is Olpidium brassicae, the first fungal vector for transmitting the virus, lettuce big vein, and it was reported by Teakle 1960. Then important characteristics about fungal transmission of plant viruses are the soil-borne nature, its soil-borne transmission, then the infection occurs through the root infection. Okay, long-term survival through fungal resting spore, because it is associated with the Jhusporangia or sporangia, so it can sustain for longer period with the fungal resting spore. Then difficult to control due to the persistent nature of fungi in the soil. Okay, the characteristics are more or less similar to the nematode transmission. Okay, next about the types of virus fungus relationship. So based on the nature as well as the retention time, the fungal and virus association was categorized into two types. First is the non-persistent transmission, and second is the persistent transmission or association. So if the virus is just carried externally on the surface of the Jhumishree, then it is non-persistent association, because the virus actually just present superficially over the Jhumishree. So it is not for the long-term retention. But in case of persistent association, the virus is retained inside the fungal Jhumishree or in the resting spore or in the cyst. So it survives the dormancy period and it provides a long-term survival for the virus in soil. So this is about persistent and non-persistent association of fungal of the virus in the fungus. So example of virus that is showing non-persistent association with fungi is tobacco necrosis virus, and the example of virus showing persistent association with fungi is tobacco stunt virus. This is stunt, okay, TSV. TSV. For non-persistent, it is TNV. For persistent association, the example is TSV. TNV, TSV, okay. Remember the examples, okay. Next, so I have already discussed in the plant virus transmission video that the two group of fungi are reported as, or two phylum of fungi are reported as vector for plant virus transmission. The Chytridiomycetes or Chytridiomycota and the Plasmodiophoromycota. Actually, the Plasmodiophoromycota, these are protozoan fungi, but previously it was categorized as under the fungi kingdom only. So we are discussing it under the fungi only, okay. So first the Chytridiomycota. So Chytridiomycetes, the key feature you know that it is having motile zoospore, and the major characteristic thing is the presence of single posterior flagellum, okay, and the virus is soil borne, and the transmission occurs through the infection of zoospore into the infected roots, okay. So examples of fungi under the Chytridiomycota that act as vector for plant virus transmission, these three are very, very important example, okay. Remember the name of the fungus as well as the name of the virus transmitted by the particular fungus. The association of this fungus and the virus is very, very specific. The particular species can transmit a particular virus only, okay. So Synchytrium endoviticum will transmit the plant virus, the potato virus X only, not potato virus Y, okay. So this the transmission is very, very specific. Then about this Olpidium brassicae, so it transmit the TNV as well as lettuce big vein virus, and Olpidium bournovirus, it transmit the cucumber necrosis virus CNV, okay. So these are the three key fungi under Chytridiomycota that act as vector for plant viruses. Very, very important, okay. You have to remember all the examples. Next about the Plasmodiophoromycota. So under this family, these are the protozoan like fungi, okay. These are the members are obligate root parasites, and in case of the Chytridiomycota, here we have discussed the zoospore are having the posterior single flagellum, but here in case of this Plasmodiophoromycota, two plus flagellum will be there. Then it produces long-lived resting spores. By the association of resting spores, this virus actually can persist for longer period. The virus are soil borne in nature, and when during the favorable condition, the resting spore will release the zoospore, and the virus or viruliferous zoospore will then infect the healthy plant and transmit the plant virus. So examples of fungi under the Plasmodiophoromycota or the protozoan like fungi under this Plasmodiophoromycota that act as vector for plant virus transmission are very important three examples, okay. Remember these three. The Polymyxa graminis, it is reported to transmit peanut clump virus and wheat soil borne mosaic virus. Then Polymyxa betae, that is reported to transmit bead necrotic yellow vein virus, and Spongospora subterranea, this is reported to transmit the potato mop top virus. Here also the transmission, the relationship is very specific. The Spongospora subterranea only transmit potato mop top virus. It can't transmit peanut clump virus. Okay, so the association is very specific in both Chytridiomycota as well as the Plasmodiophoromycota. So remember examples, these are very important and directly it will be asked in the exams. Next, about some important features. So like we have discussed, the transmission occurs through the root infection by the Viroliferous zoospore, and the viruses that are transmitted are the soil-borne nature. Then zoospore act as vector, maybe internally or externally. So both non-persistent as well as persistent transmission was seen. And long-term association or long-term survival of the virus occur by the resting spore. And it is difficult to control because of persistent nature of the fungi in soil. Because most of these fungi are soil-borne in nature, so it's quite difficult to manage. So this is about fungus as a vector for plant viruses or fungal transmission of plant viruses. I hope these things are clear to you.